And now I'm going to talk about the T lymphocyte. And as I mentioned that, the common lymphoid progenitor has a capacity to give B lymphocyte. T cells or precursor or pre-T cells, they originate from bone marrow as CD4 negative and CD8 negative, and IL-2 actually is very important for their proliferation. Then they migrate from the bone marrow to the thymus gland above the heart, and they enter the thymus gland through the hives or high endothelial venules. These are the pre-T cells. Sorry. And the pre-T cells, they arrange genes that encode beta, membrane-bound beta chain. As T cells, as you know, they express T cell receptor. It is formed of alpha and beta chain. First, they express beta chain, and this beta chain combine pre-T alpha. So the alpha is not complete. For that reason, the T cell is immature, and it is associated with CD3, and this CD3 is the marker for T cell. When the pre-T cell rearrange the alpha chain, they become pro-T cells. And now they become double positive, because when they enter the thymus gland, they are double negative. Now they become CD4 positive, CD8 positive. This CD4, this double positive pro-T cell that express alpha and beta chain interact with thymic epithelial cells within the cortex of the thymus gland. The thymus epithelial cells, they express major histocompatibility class 1 and major histocompatibility class 2. And if they interact with the major histocompatibility class 1, they will be differentiated into CD8 or T cytotoxic. But if they interact with major histocompatibility class 2, they will differentiate into CD4. And this recognition of the major histocompatibility here in the cortex is known as positive selection. And actually, during this stage, they get some education because as you know that the major histocompatibility molecules are responsible for recognition and discrimination between self and self, between self and non-self. So T lymphocyte, they migrate from bone marrow to the thymus gland. First, they for maturation, then to get education. How do they get education? They go deep within the cortex, where they meet the macrophages and the dendritic cells. These macrophages and dendritic cells, they express on their surface self-antigens in association with major histocompatibility. So they can now see self-antigens or antigens in our bodies. They get some education. So the thymus gland is school actually. If they recognize the self-antigens expressed by these cells, they will not be killed by apoptosis. But if they fail to recognize self-antigen association with self-major histocompatibility, they will be killed by apoptosis. But if this recognition, if it was by high affinity also, they will be killed. Other purpose for the migration of T cell to the thymus gland is the development of the repertoire. Development of the repertoire, it means that T cells, when they arrive the thymus gland, they divide themselves, a clone or a population sit here, and a population will sit here, and a population will sit here. They do not sit in one place. And why? The purpose behind that is that this development of repertoire, for example, this population decide to protect you against malaria in the future. This again is Yersinia, this again is tuberculosis, this again is enteric fever. So this we call it development of repertoire.